Learners, good morning to everyone. Last video we have discussed about the receptors like GPCRs. Today we are going to discuss about some kinase receptor like tyrosine kinase, JAK-STAT and serine kinase. Tyrosine kinase receptor is a homodimer type of receptor which is act by phosphorylation of proteins. How does it act? First, when a hormone binds with the tyrosine kinase homodimer receptor, this is one subunit, this is another subunit. They are similar in structure, so they called as homodimer receptor. There will be internal changes of structures, and ultimately there is changes of some bonding which lead to phosphorylation of these homodimer structures. This phosphorylation is reciprocal type of phosphorylation. This subunit will phosphorylate this one and this subunit will phosphorylate this one. After this, all phosphorylation has been done. The second messenger protein will come here and they will be phosphorylated by this homodimer tyrosine kinase receptor. This phosphorylated protein will act further and will manifest or will act as a functional protein for this hormone intracellularly. This receptor is important for insulin and other growth factors like epidermal growth factor, insulin like growth factor, nerve growth factor, platelet derived growth factor, fibroblast growth factor. All this type of growth factor and insulin act via this tyrosine kinase receptors. This is very important. There are certain drugs which ends with tinib. For example, sunitinib, bosutinib, dasatinib, nilotinib, ponatinib, regorafenib, these all are tyrosine kinase inhibitor. Tyrosine kinase inhibitor. So this drug more or less used in CML. CML is chronic myeloid leukemia. One of the major reasons for developing CML is Philadelphia chromosome. Philadelphia chromosome, which is nothing but translocation between chromosome 9 and 22 and fusion of BCR ABL gene. This is a type of tyrosine kinase receptor which is blocked by this tinib drugs and ultimately leads to downregulation of this tyrosine kinase receptor and some improvement in CMLs. Sunitinib and regorafenib also used in case of hepatocellular carcinoma. Now let's go to the next type of receptor, which is JAK-STAT receptors. JAK-STAT is nothing but Janus kinase and STAT receptor. STAT is a protein and Janus kinase is the actual receptor. Janus kinase receptor is mainly used in case of mainly act for prolactin, growth hormone and erythropoietin. This is also same type of receptor like tyrosine kinase. This is homodimer subunits, and those hormone which attaches in this homodimer protein will act through this Janus kinase and it will phosphorylate a protein which is known as STAT protein. This phosphorylated STAT protein is nothing but active form of STAT. This active form of STAT will go and bind to hormone receptor element in the DNA. hormone receptor element is a part of DNA which will further transcribed and translocated into protein, mRNA and protein subsequently, and this protein will manifest all functional responses of this hormone. So this is about JAK-STAT kinase receptors. Now the last group of receptor, which is serine threonine kinase. This serine threonine kinase receptor is more or less even similar like tyrosine kinase and JAK-STAT kinase receptor. This is also a homodimer type of receptor which mainly for TGF beta, or activin or inhibin or bone morphogenic proteins. So this homodimer will phosphorylate a regulatory protein which is known as SMAD gene regulatory protein. This will form a heterodimer. Another SMAD regulatory protein will attach with this phosphorylated SMAD regulatory protein subunit, and this heterodimer will go inside the nucleus and will attach with the SMAD gene, and this SMAD gene will then further expressed via transcription and translation and lead to manifestation of all the functional responses of this TGF beta or activin or inhibin or bone morphogenic proteins. This is very important for tissue development and differentiation. You guys know that TGF beta is for tissue development and differentiation because it is a growth factor. Bone morphogenic protein is also a type of growth factor type chemical. Mutation of this pathway lead to cancer. Why? Because this is for tissue development and differentiation. If this gets mutated, then differentiation will not happen. Immature cells like cancer cell will develop and tissue development will not also be happen. SMAD dependent transcription is the major mechanism behind this serine threonine kinase receptors. You guys have seen that here, that SMAD gene will further transcribed and translocated to different proteins which lead to functional manifestation of TGF beta or others hormones. Now another important receptor which is thyroxine receptor. We know that thyroxine majorly secreted as a form of T4. This will convert it to T3 by a deiodinase enzyme. This T3 will go through and through to the cell membrane and nuclear membrane into the nucleus, because this T3 is lipid soluble hormone. So no issue in perforation of this cell membrane and nuclear membrane. After going inside the nucleus, this will attach with the thyroxine receptor which is present inside the nucleus, and this thyroxine receptor will form a heterodimer with retinoin X receptor. This is nothing but a vitamin D dependent receptor. This heterodimer in the presence of another co-activator will stimulate the thyroxine receptor element. This is nothing but a portion of gene, and this gene will lead to transcription and translation of mRNA and protein, and this will lead to manifestation of functions of thyroxine in different tissue levels.